So yesterday I was telling you about optical methods, various optical methods that we use to measure cerebral blood flow. Today I want to start talking about measuring brain activity using optical methods. And I'm going to do that in the context of kind of addressing this question of neurovascular coupling. So with optical methods and with fMRI, I'm sure you've all heard of fMRI, you're actually measuring the hemodynamic response to brain activity. You're not measuring the neural activity directly, right? And so an interesting question is, what is this relationship between the neural activity, you know, the action potentials and such, the electrical wiring, and the blood flow response? And so actually the work I'm going to be describing to you is principally all done by Anna DeVore, who I've had the pleasure of working with for the last 20 years. She's presently now at University of California, San Diego. But all of this work I'm going to describe to you is done a long time ago, actually, at Mass General Hospital. And so, I mean, this diagram here is basically what I described to you. There's neural activity, so action potentials in the axons, synaptic release of neurotransmitters, you know, propagate to the neighboring neurons, and it releases vasoactive agents. They diffuse to the blood vessels and the smooth muscle cells on the arterioles, which relaxes them, and so then they dilate. And as they dilate, the vascular resistance diminishes and blood flow increases. And that blood flow increases, delivering more oxygenated hemoglobin to the capillaries to support, presumably, the increased metabolic demand needed to repolarize the neurons, right? And so a question is this neurovascular coupling, is this a feedback mechanism to sustain the metabolic demand for oxygen, or is it a feed-forward mechanism anticipating increases in oxygen consumption? Right, so this was debated for a long time. And so I'm going to describe to you results we have making measurements actually from across scales, invasive measurements in animals to non-invasive measurements in humans. And I'm going to be describing to you the use of many different optical methods, from multiphoton microscopy, which I did not talk about yesterday, I believe, to the laser speckle imaging that I did talk about yesterday, and near-infrared spectroscopy measurements in humans. Okay, so we're going to be looking at the neurovascular spatial relationship, the role of excitatory and inhibitory neural activity, and the impact on the vascular response and the impact on the metabolic response. And then we're going to translate to humans and see if what we learned in animal models also holds true in humans. Okay, so starting off, we were using intrinsic signal, optical signal imaging, and laser speckle contrast imaging. So I told you about laser speckle contrast imaging yesterday. We just shine a laser beam on a sample of interest, and we measure the speckle pattern that comes back. And because of moving blood, because of blood flow, that speckle pattern is fluctuating in time. And if we integrate that fluctuation over the exposure time of a camera, we get a reduction in speckle contrast that's proportional to the amount of blood flow. And so that way we can monitor changes in blood flow during neural activity. That's easy enough. We combine that with multispectral imaging. So hemoglobin, you know, absorbs very strongly in the blue and green and less strongly in the red. So that's why if you shine a flashlight on your hand, you see red light transmitted through your hand. It's because of all the blue and green light is absorbed by the hemoglobin. And the hemoglobin absorption depends on whether or not oxygen is bound to hemoglobin. So using different wavelengths of light, we can probe the relative amounts of oxygenated hemoglobin to deoxygenated hemoglobin. And so in this experimental setup, we're actually using 6 different colors of light here, probing. This is the absorption spectrum of hemoglobin you see here, and so we can probe different wavelengths of light are more or less sensitive to oxy or deoxyhemoglobin. Okay, and so using this intrinsic signal imaging, if we do, in this case, this would be a forepaw stimulus, so we're giving electrical shock to the forepaw of a rat, and it produces a sensory response in the cortex, and we can image that sensory response by looking at the changes in hemoglobin concentration that you see here, depicted here. We could also then go in with 2-photon microscopy We can image these blood vessels. We basically put a fluorescent dye into the vasculature. And so that it fluoresces the vasculature. And because you have micron resolution with two-photon microscopy, we can measure across these blood vessels and during neural activity, see how the diameter of those blood vessels changes, right? Because the release of vasoactive agents from the synapses are diffusing to the arterioles, relaxing the smooth muscle cells, and they dilate. So we can actually measure that dilation of the arterioles directly with two-photon microscopy, okay? So this was an early result that Anna DeVore got, a very elegant result, kind of the highest contrast-to-noise ratio measurement really ever made of this vascular response to neural activity. She was actually deflecting the whisker on a rat, a single whisker and a single deflection. And so that gives a very clean electrophysiological response over 100 milliseconds. Basically, you know, that sensory stimulus produces a neural response in the brain that's over and done with within 100 milliseconds. And then if you average enough, I think she averaged 1,000 trials here, you can see what the hemodynamic response is, right? So 100 milliseconds, everything's done. And then a few hundred milliseconds later, you can begin to see an oxygenated hemoglobin response, a deoxygenated hemoglobin response that builds over time, right? And so it builds over seconds, right? So this is thousands of milliseconds or seconds, and kind of the peak response is occurring 2 seconds after the stimulus, right? So the neural response is done with in 100 milliseconds, but the vascular response takes 2 seconds to peak, right? And so this is a very typical response. We get an increase in oxyhemoglobin, this delayed decrease in deoxyhemoglobin. So what's going on here is, I mean, the story is simple. Let's just look at blood volume. So remember, we have a neural activity. It releases vasoactive agents, diffuses to the blood vessels. They dilate. When they dilate, it's going to increase blood volume. So basically, this total hemoglobin concentration here is the measurement of blood volume. It's the measurement, essentially, of the arterial dilation, right? So this arterial dilation takes about a second before we actually begin to observe it. You know, it peaks at about 2 seconds. Before this arterial dilation happens, we actually do see some oxygenated and deoxyhemoglobin changes, right? So what's happening here is basically we're seeing an increase in oxygen consumption, right? So the neurons are already trying to repolarize. They're already increase their metabolic activity. They're already consuming more oxygen. And we can see that because oxyhemoglobin decreases and deoxyhemoglobin increases. But when that blood volume increases, what happens is blood flow increases, right? Because we reduce the vascular resistance, blood flow increases, delivers more oxygen, and actually it delivers more oxygen to the tissue than is actually needed, and that's why we see that this oxyhemoglobin increases, right? So I hope that makes sense. It's interesting. The blood flow response always oversupplies the oxygen need of the tissue. And actually, there's really interesting reasons why for that, which I don't get into in this talk, but tomorrow I'll get into it. Okay, but if you look closely at these data, you know, surrounding the positive response here, you can see a negative response. And here you have a decrease in deoxyhemoglobin. Surrounding it, you can actually see an increase in deoxyhemoglobin. So there's this surround negativity. And the question I'll come back to is that a passive response or is it an active response? Is there actually some active neural activity producing that surround negativity? We initially thought actually it was a passive stealing of blood, that more blood was needed in that center of brain activity to support the metabolic demand, and so we thought blood was being stolen, and that was a passive phenomenon. Okay, but let's just focus for a few slides on the center of brain activity. So this is Anna. Anna actually did multiple different amplitudes of the stimulus. So she could flick the whisker 10 microns, 20 microns, 100 microns, and the more you deflected the whisker, the bigger the neural response you'd get. Right. So this is showing the amplitude of the hemodynamic response for different stimulus amplitudes. And at the same time, she had electrodes in the brain, and she could measure the neural activity. She could measure the multi-unit activity. This is the spiking activity of the neurons, and she can measure the local field potential. This would be really an aggregate average of the depolarization of the neurons. Okay, and by doing this parametrically with many different amplitudes, you could then begin to look at the coupling between the hemodynamic response and the stimulus amplitude. So this we see is very linear. As the amplitude of the stimulus got larger and larger, the hemodynamic response got linearly larger. If we look at the multi-unit activity in the local field potential, we see that actually it saturates, right? They're linearly related to each other, but the relation to the stimulus amplitude is actually saturating, right? So this implies that there's a nonlinear relationship between the neural activity and the vascular response, right? And this was surprising to us, that you'd have this saturation of neural activity, but the hemodynamic response would keep going up. You know, how could that happen? Well, I need to tell you, the Whisker barrel cortex has a very interesting structure. So a mouse has many whiskers, 20 whiskers about, and each whisker is mapped to the cortex in a very organized pattern. So she was just deflecting a single whisker here, which corresponds with a barrel of neurons in this location, right? But surrounding it are barrels of neurons that are coupled with all of the other whiskers. And so we surmised, hypothesized that basically what's happening is the vascular response is spatially integrating the neural response from all of those barrels, right? And so we were just here looking at the neural activity in a single barrel. But so we had to go and measure the neural activity in the surrounding barrels. And when we did that, we saw that while that principal barrel was saturating, the surrounding barrels were still showing linear increases in their neural activity, right? So basically, this was confirming for us that this vascular response, is not measuring the neural response from a single barrel of activity. It's actually spatially integrating the neural activity from many of those barrels, right? So this is telling us something about the spatial length scales that the hemodynamic response is sensitive to versus the electrophysiological responses, right? So these electrodes are measuring maybe the neural activity in, say, a 100 micron area, whereas that vascular response is really integrating it over hundreds of microns, right? So we have to keep in mind that it's not just the hemodynamics has a different temporal response, but it also has a different spatial response. Okay, but let's come back to this surround negativity, right? Is this surround negativity passive or is it active, right? So we did some modeling, and this was a lot of fun. I like to do modeling. So at this time, people had been modeling the hemodynamic response to brain activity just by having essentially a series of pipes. You'd have an arterial that could dilate, and then you'd have a capillary that could passively respond, and a vein that could passively respond. A very simple linear serial network of pipes. But in fact, the microvascular network has a complex topography. We could simplify it, but make it a little more complicated by considering the branches of arterioles that keep branching into smaller and smaller vessels, into the capillary network and the veins. And then we could say, well, during this neural activity, I'm actually just dilating a single arteriole. And if I dilate that single arteriole, our hypothesis was it's going to increase blood flow here, and actually it's going to steal blood flow from the surrounding territories because the vascular resistance here is diminished. And so that decrease in blood flow should create this surround negativity that we're seeing in our experimental data, right? So that's how we simulate blood flow. It's just a network of resistors. It's very easy to simulate. Oxygen delivery, basically you just have oxygen convecting through these pipes, these blood vessels, and then diffusing into the tissue where they're consumed, right? So it's a fun mathematical model to set up, and we could solve it just by dilating the local arterioles at this level. You can see increase in blood volume. This is 200 milliseconds during the dilation. You get this local increase in blood volume. Very rapidly, you see the blood flow increase there downstream from those dilating arterioles. You could see in the surrounding blood vessels, actually blood flow has decreased. And as we look longer and longer in time, as that blood flow increase propagates downstream, we see the oxygenated hemoglobin, this is the oxygen saturation of hemoglobin, is increasing downstream of the dilating arterioles, but in the surrounding region, it's decreased, right? Because blood flow is decreased. So this was why we were arguing that that surround negativity is actually a passive stealing of blood to the active region, right? So we could then we could take this model and fit our experimental data. So this is the temporal, the symbols are the experimental data, and I just take from the theoretical model at a region of interest from the center of activation. We could get a fit to the data very well. I could then take a region of interest in the surrounding territory and see how well it matched our experimental data, and basically there is a poor match. So the theoretical prediction has a surround negativity, but it's much smaller than what we experimentally measured. And there was nothing we could actually do to this model that would allow us to predict the large surround negativity that we experimentally observed. So that suggested to us there must be active mechanisms that are driving this hemodynamic surround negativity. And so we had to figure out what that was, because our initial measurements with electrophysiological measurements and multi-unit activity, local field potential, wasn't revealing to us any active neural process that could produce that surround negativity. So these are the hemodynamic responses again. What Anna then did was set up a voltage-sensitive dye recording measurement. So she had dyes that would embed into the membranes of the neurons, and their fluorescence would be modulated by the membrane depolarization, so the potential difference across the membranes. So in this way, she could map out spatially temporally the changes in the neuronal membrane potentials. And I mean, so this is happening not over a seconds time scale, right? It's happening over a tens of milliseconds time scale. So at this time when we were doing the measurements, you needed really fancy cameras that could image fast enough. And so this is showing the images every 5 milliseconds. I mean, today this is kind of routine, but 15, 20 years ago, this was really cutting edge technology. So we see the single Whisker deflection. You can see increased fluorescence. This is an indication of depolarization of the neurons. And then after about 100 milliseconds, it's returned back to baseline, and actually it undershoots. So this is now a hyperpolarization of the neurons, and this is always happening. And then she looked at these, she made these circles, right? And so you could look at the activity in the center and in the surround. And so here's oxyhemoglobin in the center, and as you go radially outwards, you see it's diminished and diminished, and actually in the surround, you just see that it's this negativity. So you could do the same thing with the voltages, the membrane potential measurements, and you see in the center a strong depolarization followed by some weak hyperpolarization of the neurons. As you go to the surround, there's actually weak depolarization and even stronger hyperpolarization, right? So there's more hyperpolarization occurring in the surrounding neurons, right? So we began to get a hint that actually this is looking like the surround negativity. Maybe it's this hyperpolarization here that is greater in the surrounding territory that's driving this extra vasoconstriction, this decrease in blood flow that we're observing in the hemodynamic response, right? So she wanted to get more evidence of that. She went in with 2-photon microscopy and could measure in the center of the neural activity here these clear diameter increases, and then going to the surround, you could begin to see there would be weak dilation of the arterioles followed by a strong constriction of the arterioles, right? So something's driving this strong constriction, and we're hypothesizing that it's this surround hyperpolarization of the neurons, right? So we created a very simple model of the arterial diameter changes. You could say there's a dilation component and a constrictive component, and just by taking a linear combination of the two, we could fit our experimental data, right? As you see here, here's some examples of our fits to the experimental data. We get the coefficient for dilation, a coefficient for constriction, And basically, we look at the ratio of those coefficients. As we go further and further away from the center of brain activity, we see that there's relatively more constriction of the arterioles, right? And we basically, are able to relate that to the relatively greater amount of hyperpolarization. And this hyperpolarization is happening because of neuronal inhibition, right? So it allowed us to kind of develop this very simple model that depolarization is releasing vasodilatory agents that diffuse to the smooth muscle cells, causes them to dilate, whereas hyperpolarization, which is coming from inhibitory neurons, those inhibitory neurons are releasing vasoactive agents that actually cause the arterioles to constrict, right? And so actually now recently, and I'm not going to show you the recent data, recently Anna's actually been able to go in and optogenetically specifically activate the inhibitory neurons versus the excitatory neurons, and can basically see that it's the only way to get any vasoconstriction in those arterioles is by exciting the inhibitory neurons, and specifically inhibitory neurons that release neuropeptide Y, NPY, a very specific type of vasoactive agent that is causing the arterioles to constrict, right? Okay. But Anna ran, when do I finish? I forget. 11:10, yeah, okay. Okay, so we still have this question of, there's this surround negativity that would, the blood flow is decreasing, that would suggest metabolism is decreasing if we have a feedback mechanism of the blood flow response to support metabolic demand, right? So if blood flow is decreasing, it must be that metabolism is decreasing. But we see increased inhibitory activity. So that'd be strange. How could there be increased inhibitory activity, which would be more energy consuming, but blood flow is going down, right? So we wanted to confirm our suspicion that we had this decoupling between energy demand and blood flow, which would kind of mean that this vascular response is not feedback, it's kind of a feed-forward mechanism. But we couldn't do it with the data set I showed you, so we had to create another experiment. And in this experiment, it was a forepaw stimulus, and we're looking at, so you stimulate, say, the left forepaw, you're going to activate the right hemisphere, but there is neural communication between the hemispheres. And so in this experiment, we looked at the two hemispheres of the rat, forepaw region, and in the contralateral hemisphere to the stimulus, we see that local increase in blood volume responding to the stimulus, diminishing over time. We also have the speckle contrast measurement. Speckle contrast decreases when there's an increase in blood flow. We see that in the contralateral hemisphere. You can actually see the surround negativity as well, where actually blood flow is decreasing. But now we look at the ipsilateral hemisphere, and we actually see that blood flow is really just speckle contrast is increasing, which means blood flow is decreasing, right? So in the ipsilateral hemisphere, we're getting this decrease in blood flow, and we know that actually when you stimulate one hemisphere in the sensory cortex, there's inhibitory activity going to the other hemisphere. So this decrease in blood flow is consistent with everything I was telling you. There's increased inhibitory activity here, so there's decrease in blood flow. But we hypothesize there must be an increase in energy demand even though there's this decrease in blood flow. And we were, this is just showing figures of that, right? So, well, just to complete the story, Anna did the voltage-sensitive dye imaging, and really the same thing. In the ipsilateral cortex, you could see there's less depolarization, but even more hyperpolarization. There's more inhibitory activity, right? So, and she did all the two-photon microscopy measurements of the dilation, and we see that the ipsilateral cortex is constricting, right? So, we're expecting that there's increased metabolic activity, but blood flow is decreasing, right? So there's this discoupling between flow and consumption. At this point, everybody in the fMRI community was saying blood flow is linearly coupled to the neural response, right? So here we're able to, and that would be a feedback mechanism. It needs to feed back to the energy demand. But here we've been able to trick the system and break that relationship and suggesting that would be more feed forward, and I'll explain that in a minute. Excuse me. And to do that, we had to measure metabolism. And so we were lucky to work with Christian Weber, who used a 2D radiographic method to measure glucose uptake. So you can feed your mouse or your rat a glucose, a radioactive glucose, and during brain activity, that glucose is going to be consumed by the neurons, and then you can remove the brain and measure the radioactivity of slices of the brain and see that increased metabolism. And so here you can see, this is the contralateral cortex, you can see an increase in glucose uptake here, which is an indication of increased glucose metabolism. And so we were able to measure at many different places, and you can see in the contralateral hemisphere increased glucose consumption, but also in the ipsilateral cortex we had increased glucose consumption. Right? So basically the conclusions are that functional activation is predominantly related with active dilation of the arterioles, but there's also inhibitory signals that are driving a vasoconstrictive response. This actually allowed us to create an experiment where we were able to decouple the blood flow response and the neural response and get the system to reveal that it's really a feed-forward mechanism. So basically what's happening is, we did a very short stimulus where there's increased inhibitory neural activity, which is more energetically demanding, but it's feed-forwarding a blood flow decrease. And the reason it's doing that is because if you sustain this stimulus of inhibitory signaling, actually the neural activity is going to decrease. And so sustained for several seconds, the neural activity will decrease and the energy metabolism will decrease, right? But because we only did a very brief stimulus, we were able to kind of fool that system to reveal that it's actually a feed-forward mechanism. Okay. So how do we take these findings and bridge it to humans, right? We can't actually open up the skull very easily on a human and put electrodes in the brain. I mean, during some surgeries you can do that, but you don't have much time. So we wanted to do this with non-invasive measurements, and so we combined near-infrared spectroscopy, which is measuring the hemodynamic response to brain activity, with magnetoencephalography, which is measuring the neural response more directly by measuring the magnetic fields, right? And so Mariangela Franceschini was able to do this study where she was able to put the fiber optics for measuring the near signal and the hemodynamic response onto the subject, and then put the subject into an MEG system, right? So maybe Arjun told you about near-infrared spectroscopy. I'll just give you a recap on it. So we're going into the near-infrared, where hemoglobin absorption is even less, and so light is able to penetrate centimeters into tissue. And by using 2 wavelengths or more, we can probe changes in oxy and deoxyhemoglobin concentration. And we do this by putting sources and detectors on the head 3 centimeters apart. The light we detect is sampling the cortex, so any absorption changes in the cortex is going to modulate the amount of light we receive at the detector. And we can do this with good temporal resolution, so we can measure the temporal response to neural activity. So this is showing the positions of all the sources and detectors on the brain, and underneath here is actually the MEG response to a sensory stimulus. So we're giving an electrical shock to the median nerve, which is producing a sensory response. This is showing the neural response, and then overlaid on that is the hemodynamic response. And you can see good spatial correlation between the neural response and the hemodynamic response. So we can look at these temporal responses. This is looking at deoxyhemoglobin. And we did 1 second duration stimulus, 2 seconds, 3 seconds, 4 seconds. So basically it's a 3 hertz stimulus to the median nerve, and you can either do that for 1 second, 2 seconds, 3 or 4 seconds, right? So here's the response. And so one question is the hemodynamic response linear? So one way you can explore that is you could take the 1 second response, convolve it with a 2 second duration stimulus, and see how well it predicts. And basically we see our prediction doesn't match well the observation, right? So suggesting there's a nonlinearity. If instead we take the response to a 2 second stimulus and predict the 4 seconds, we actually get a very good prediction, right? So it suggests that there's some nonlinearity in response to short stimuli, right? So this is another way of looking at the amplitude of the deoxyhemoglobin response. You know, for 2, 3, 4 seconds, it's linearly increasing, but for a short stimulation, you know, there's this nonlinearity, right? This was actually also observed with fMRI. Peter Bandettini showed this, and in the fMRI literature, there is this debate. Is this nonlinearity a nonlinear hemodynamic response, or is it just reflecting a nonlinearity in the neural response, right? So this was a good question. At that time, you know, you couldn't do fMRI and MEG at the same time. You know, fMRI is 7 Tesla, MEG is femtotesla. You know, something about 15 orders of magnitude difference makes it hard to make those measurements simultaneously. But, you know, the fiber optics and near-infrared spectroscopy, we could do it simultaneously. So we could answer the question, is this nonlinearity, right, really just a nonlinear neural response to the stimulus, right? We know that that is the case, but it's still the question, is the coupling from the neural signal to the vascular signal, is it linear or is it nonlinear, right? Okay, and so to do that, you can look at the temporal responses to the 3 hertz, or actually it's a 4 hertz stimulus, and this is the MEG response, and you can measure the stimulus onset is there, and then over 100 milliseconds you see the response. You have a negative response at 20 milliseconds, a positive response at 35 milliseconds, so on and so forth, and these are actually reflecting different components of the neural activity as it goes from the median nerve to the thalamus, and then from the thalamus out to the cortex. We can actually resolve all of those different temporal components. Now, to do this, answer this question of the response, is it linear or nonlinear, we can just do a regression analysis. So you can measure the area under the curve of each of these components and then see how it predicts the hemodynamic response. And so there's the hemodynamic response. And if we just take the N20 response from MEG, you know, it doesn't predict the hemodynamic response very well. You could take just a positive 35 millisecond response, you know, or the P60, or the total. And basically, when we did this analysis, we found that we had to integrate the full electrophysiological response in order to get a good model prediction of the hemodynamic response. Okay. So this kind of confirmed for us that there is a linear relationship between the neural response and the hemodynamic response. So we like that, right? It means that with fMRI and near-infrared spectroscopy, when we're measuring that hemodynamic response, it's a linear representation of the underlying neural activity. And so any nonlinearities you observe in the hemodynamic response are actually going to be a reflection of the underlying nonlinearities of the neural response to the stimulus, right? So that's really important to know that. And the other thing that came out of this is that we really needed those later neural responses to predict the hemodynamic response, right? So let me just explain that a little bit. The early neural responses are the thalamic inputs into the cortex. Once those thalamic inputs reach the cortex, actually layer 4 of the cortex, then you get a widespread neural activity across the cortical region, across the layers. And that's happening at these later time points. And there's much more synaptic activity in the cortex happening at that time. And it's really nice to see from these data that you have to consider all of that neural activity to predict the hemodynamic response. At that time, there had been some high-profile fMRI papers where they, in monkeys, had electrodes measuring essentially the thalamic inputs. And they showed that they could predict the hemodynamic response. And they were kind of implying that the hemodynamic response was related to the thalamic inputs. Well, that's true. Without the thalamic inputs, you don't get a hemodynamic response. But to actually predict a hemodynamic response, you need to fully understand the spatial integration of all of the neural activity in that cortical region. Right. So to kind of delve more deeply into that, Mariangela Franceschini did really nice studies in rats where she was able to tease apart the neural responses in a much more detailed fashion. But I'm not going to have time to tell you that, so I'll just jump right to the end, to the summary, and would be really happy to entertain any questions. Yeah, thank you.
Questions?
Actually, you may expect the vasoconstriction in response to the vasodilation in order to maintain homeostasis. And as we are using it for practical destruction of the tumor, once you deliver the nitric oxide for the vasodilation, concomitantly you will have a delivery of endothelin-1 or the other constrictor that you are having. And usually they have a longer lifetime in the vasculature than the vasodilators.
Yes.
And therefore you may be staying with the vasoconstriction. But I'm not sure that you measure really the overall area, because the vasoconstriction of one part can lead to an opening of an intermittent blood flow within non-functional blood vessel before the stimuli. So it could be that this non-functional blood vessel prior to the stimuli will now become functional. So it is actually three-component system that you may have to look at.
Yeah, I mean, so our modeling showed that for sure there is a passive stealing, right? But it's a minority response. The majority response is that active vasoconstriction from the inhibitory signaling. There are functional collaterals, and people have shown that effect very nicely actually in stroke models. And it's interesting, rats have functional collaterals, but mice do not have functional collaterals. And so that's kind of interesting to try to understand why that is. Humans probably have functional collaterals, and it's actually a very good mechanism to help your brain persevere in the presence of any occlusions, like a stroke, right? So that's very important. The long-lasting nature of the vasoconstrictive signaling, right, is in fact a key component, I think, of this feed-forward vascular response of neural activity, right? Because if you have repeated inhibitory signaling, basically you're setting up so that down the road over seconds, minutes, or whatever, there's going to be reduced neural activity. And so you need a long-lasting effect of reduced blood flow. And so that's exactly, I didn't have time to show it in all the data, but our data shows that the vasoconstrictive signaling is much more long-lasting.
About 20 seconds per each pulse.
Yeah, cool.
The vasoconstriction.
How do you know all that? That's great.
And 11 to 13 seconds for the nitric oxide.
Fantastic. We have to talk more.
Hi, it's a nice talk. Thank you.
Our data is consistent.
So,
with that, right?
Your neuronal response. How do you measure neuronal response? Is
I
a
can't
voltage?
really hear you. Sorry.
Sorry. Do you measure voltage or current for the neuronal response?
It's measuring voltage, the membrane potential, right? So the membrane potential changes causes conformational changes in the fluorescent dye, which changes the fluorescence.
So maybe the blood flow will be proportional to energy, because the energy will go with the square of voltage, and that will be nonlinear.
No, I don't understand. Sorry. So
it's power, the power consumed.
So the last results I skipped over, was kind of going into great gory detail of all of the components of the thalamic inputs versus the intracortical processing, and from the electrophysiological measurements, doing a linear regression with the hemodynamic response. And basically, in the human, we weren't able to tease apart the inputs from the outputs, essentially, the intracortical processing. But in the rat model, we were able to do that, and really, the inputs really we didn't need the inputs at all to predict the hemodynamic response. Just measuring that intracortical activity, we could very nicely and very accurately linearly predict the hemodynamic response, right? So all the evidence is there's a strong linear relationship between certain components of the neural response and the hemodynamic response.
All right, thank you.
Yeah.
More questions? So let's thank you again, Professor David Boas.